नमस्कार दोस्तों, आप देख रहे हैं साइंटिस्ट आर एकेडमी टूवर्ड साइंटिफिक वर्ल्ड. Subscribe to my channel for more scientific videos and click on the bell icon for updates. In this video lecture, we are going to understand the different sources for the endophytes isolation. We know that endophytes are the very critical organism present in the environment, and therefore we have to go for the such a good sources those we can isolate the good endophytic fungi or good endophytic bacteria from the sources. So in this video lecture we are going to discuss different sources for the endophyte isolation and their research. Let's start with first one, that is grass. Sido in 1914, Balancia Anthropogenicidea and H.E. Butler from Andropigna acuata. The first scientist Sido, he isolated endophytic fungi from Andropogon acuata. Next one, after 15 years, Govindu and Thirumalchar in 1964, they have isolated endophytes from the grass. Total grass species, that is 25 to 30%, are endophytes which can play a role in plant society. Based on the findings from microscopy examination, C.R. Rodriguez et al. 2007 explained the growth of Ecopy and Neotypidium endophytes in the host grasses. According to Skulls in 1998, vascular bundles of Neotypidium endophytes contain few hyphae in natural associations with grasses. In 1997, Janardhan and Ahmad reported Balancia and its anarchic state ephelis from various grasses from Karnataka, Bihar, Uttar Pradesh, Tamil Nadu. Now see here, grass is one of the important source for the endophytes isolation, and different scientists had been worked on that. First, Sido worked on the grasses in 1940, and after 50 years, the Govinda and Thirumalchar worked on the endophytes from the grass. We know that there are different types of endophytes, but from the grass, total of 25 to 30% endophytes are purified till date. And different scientists explain the role of endophytic fungi or endophytic bacteria in the different types of applications. Next is the plants. Nowadays, medicinal plants are under limelight due to their attention as they produce novel secondary metabolites against different pathogens. A number of medicinal plants have been studied for the purification and their therapeutic activities. Mainly Trigonolaph, Fuinum gracium, Silbillum marmium, Allium sativum, Allium sipa, Momacardia championus, Camellia sinensis, Azentricata indica, Catharanzus roses, Pathrium hastropsis, Ficus religiosa, Coffea arbicia, Nothabras pneumonia, Pongamia, Ashwagandha, Taxus brevilla. There are plenty of plants from the fungal endophytes or fungal endophytic bacteria are isolated. These plants were also used for the study of fungal endophytes by many researchers. They reported anti-tuberculous activity of isolate against microtuberculosis by agar diffusion method. Explants such as roots, stem, mineral bark, twigs, leaves, fruits, and flowers of plants are used. Now, see, plants are very important and they are in the limelight due to their medicinal properties, and that's what many researchers have worked on the different plants for the isolation of endophytes, that may be endophytic fungi or endophytic bacteria, and they have proved that this bacteria or fungi, that is endophytes, are very important in the view of therapeutic applications, and mainly the explants such as roots, stem, mineral bark, twigs, leaves, and fruits and flowers are used for the isolation. Next is the mangroves. Now, mangrove is a shrub or small tree that grows in a coastal saline or brackish water, and the reports from the India for the mangroves are very rare because there is no such research on the mangroves for the endophytes isolation. Arnold A reported endophytes from Acanthisa licifolis, that is Arthrocenium, Pseudomartina, Drumerta, Sesinium, Portulastrum from mangrove plants. Clay reported cell wall enzymes such as pectate, translaminase, pectinase, and protease from endophytes and their leaf litter degradation activity when exposed to extreme conditions in terrestrial and marine environments, such as high temperatures at the tropical areas, elevated hydrostatic pressure, low temperatures in the deep sea, and elevated hydrostatic pressures, etc. Mangrove plants have shown to adapt to anaerobic soil, muddy, saline waters and brachistidal activities. Now see, this is one of the important source for the isolation of endophytes, that may be endophytic bacteria or that may be endophytic fungi. As far as the reports are concerned, there are like mere reports from India. So one should go for the mangroves for the isolation of endophytic fungi or bacteria. Thank you.